So, imagine for a second that you are, building a house. You've got all the lumber, you have the bricks, the cement, but you are completely missing the general contractor.
Oh, right, the one who actually reads the blueprints.
Exactly, the person who tells the electricians where to run the wiring.
Oh.
And, without that contractor, the house might technically still get built, but the wiring is going to be completely faulty, the plumbing might leak, and the foundation is going to be shaky.
That is honestly a perfect analogy for what is happening right now in the infant gut.
Right, because we tend to think of newborn babies as these pristine blank slates. But biologically speaking, they are an active construction site. And for a really long time, we thought we knew exactly who the general contractor was. We thought, we could just buy a replacement at the local pharmacy if the original guy didn't show up.
But today we are doing a deep dive into a study that basically says, hold on, you've got the wrong guy entirely. And the guy you actually need, he might already be extinct.
It is such a massive wake-up call. We are looking at this incredible paper published in Cell. It's from February 2026 by Xiao and colleagues, and it's called A Global Genomic Atlas of Bifidobacterium infantis.
And honestly, I don't use this word often, but this paper fundamentally rewrites the textbook on early life biology.
And when we say rewrites, we really aren't exaggerating here. It's not just a minor update to the literature. It is a massive genomic atlas. They analyze 4,098 genomes. That includes both physical isolates and what they call MAGs, or metagenome assembled genomes.
Right, from 48 different countries. And the scale here is really what makes this so definitive. Because historically, microbiome research has been, well, incredibly Western-centric.
Yeah, we've basically only studied babies in places like Boston, Berlin, or London.
Exactly. But this study completely flipped the script. They increased the data coming from low and middle income countries, or LMICs, by a factor of 12 to 17. It is a 12 to 17-fold increase in global data.
So instead of this highly skewed industrialized view, we are finally seeing the actual biological picture in places like Bangladesh, Kenya, and Malawi. And the mission of our deep dive today is to unpack exactly what they found. Because it turns out the scientific community has been fundamentally confused about two major bacteria, Bifidobacterium infantis and Bifidobacterium longum.
Confused is definitely putting it mildly. The study proves they are distinct species. There are not just slight variations of the same bug, and more alarmingly, it shows that the good bacteria we think we are losing in the modern world might actually be functionally extinct in the West.
Okay, so let's unpack this step by step. We have a taxonomy crisis, we've got a missing microbe mystery, and a probiotic industry that might actually be selling us lab rats instead of wild wolves. So let's start with the identity crisis. Who are these bugs, really?
So historically, researchers just lumped these two bacteria together. If you read the older literature, you would constantly see references to the B. infantis longum complex, or BIL for short.
BIL, right. It was just a convenient bucket to throw them into because, they looked similar under a microscope?
Pretty much. They are both bifidobacteria. They both show up in the gut. But this new study used high-resolution phylogenetic analysis to prove once and for all that they are totally different. They used a metric called average nucleotide identity, or ANI.
Let's drill down on ANI for a second, because for you listening, it's important to know how we define a species now. What exactly is that measuring?
You can think of ANI as basically a DNA paternity test for bacteria. It measures exactly how much of the genetic code is identical between two different organisms. And the scientific rule of thumb, the gold standard threshold, is 95%.
Meaning if two bacteria share less than 95% of their DNA sequence, they are definitively different species, period.
Exactly. And when they ran the numbers on the BAL complex, they fall completely outside that 95% overlap. They are clearly unequivocally distinct species. You have Bifidobacterium infantis, which we'll just call BI from now on, and Bifidobacterium longum, or BL.
BI and BL. But there's a really fascinating evolutionary twist here that the paper highlights. BI is actually the ancestral species, right?
Yes. BI is the grandparent, essentially. It is the ancient, highly specialized microbe. And over thousands of years, BL actually evolved from it to become a generalist. And we can see this written clearly in their genomes.
Because BI has a larger genome, but it is full of what scientists call pseudogenes, broken genes.
Right. And people usually think a broken gene is a bad thing.
Yeah. I mean, if I have a broken engine part in my car, the car doesn't run. Why would a bacteria want broken genes?
In evolutionary terms, it's actually a brilliant strategy. Think of it like a hiker who is preparing for a very specific, incredibly difficult climb up a single mountain. To survive that specific climb, you need to shed weight.
So you throw out the heavy camping stove, the bulky tent, the extra clothes.
Exactly. You throw out everything except the specific climbing gear you need for that one rock face. That process is called genome reduction. BI is shedding the metabolic genes it doesn't need because it is adapting strictly to one very specific environment, the infant gut. It is becoming a hyper-specialist.
I love that analogy. So the broken genes aren't a failure. They were actually evidence of absolute commitment. The bacteria is basically saying, I am never going to live anywhere else except a breastfeeding baby's gut, so I just don't need the tools to survive out in the wild.
Precisely. It burns the boats. Whereas BL, the generalist, it kept its toolkit broad. It didn't throw away the camping stove. It can survive in a lot of different environments, which makes it much more resilient, but way less specialized.
Now, the study didn't just split BI and BL apart. It also found some new family members hiding within the BL group, didn't it?
It did. So under the B. longum umbrella, we have the classic subspecies longum. Then we have subspecies sus, or BS. Now, sus literally translates to pig. For a long time, we thought this was only a bacteria found in livestock.
But I'm guessing since we were talking about human babies, that wasn't the whole story.
Not at all. They found BS in human infants too. But the real shocker was a brand new subspecies they discovered. They are calling it B. longum subspecies leicesterensis. We can call it BX.
BX sounds mysterious. Where exactly was BX hiding all this time?
It was found almost exclusively in Western populations, so the UK, the Netherlands, the US. But unlike the other bifidobacteria that live deep down in the colon, BX was popping up in breast milk and in the oral cavities of babies. It seems to hang out specifically in the mouth and throat.
Okay, so we have a brand new Western-specific mouth bug. That is wild. Let's do a quick checkpoint and recap for you listening before we get into the really heavy stuff about the extinction of these microbes. What have we covered so far?
First, the old BIL complex is dead. We have two distinct species. BI is the ancestral specialist, and BL is the evolved generalist. Second, that separation is based on the strict 95% ANI genetic rule. Third, BI is undergoing genome reduction, shedding genes to become the absolute perfect companion for a breastfeeding infant. And finally, we just discovered a new subspecies, BX, living in the mouths of Western babies.
Perfect. So that is the who. Now we need to talk about the where, because this is where the data gets genuinely shocking. We mentioned earlier that they compared data from low and middle income countries, the LMICs, against high income countries. What was the actual difference in BI prevalence?
It is literally night and day. If you look at infants in non-industrialized settings, places like rural Bangladesh or Malawi, BI is the undisputed king. It is utterly dominant.
How dominant?
The first 6 months of life, it has about an 80% prevalence. It is the primary foundational colonizer of the gut.
80%. That is a massive majority. But then we look at the industrialized West.
And it is nearly extinct. In the first 2 months of life, which is that absolutely critical early window for training the baby's immune system, BI is detected in only 1.6% of Western infants.
1.6%. I mean, that is effectively zero. We are talking about an entire generation of babies in the West growing up completely without this foundational bacteria.
It does eventually show up later in the first year for some, but only at very low levels. In the West, it has been almost entirely replaced. BL, our generalist friend, has taken over as the primary colonizer. But remember, in those non-industrialized settings, BL is just a secondary background player.
This is the missing microbe hypothesis happening right in front of us. We have fundamentally altered the internal ecosystem of our children. Why exactly is BI disappearing in the West?
It is a combination of industrialization factors: widespread use of antibiotics, high rates of C-sections, formula feeding instead of breast milk, and incredibly high levels of ambient sanitation. All these things systematically destroy the very fragile ecological niche that BI needs.
Because BI is the hyper-specialist. It threw away its camping stove. It can't survive if the conditions aren't perfect.
Exactly. And if you disrupt the transmission chain, say the mother doesn't have BI in her gut to pass down during birth, then the baby doesn't get it, and the lineage dies there. It relies entirely on vertical transmission, mother to child.
Which brings up something incredible this study found about this transmission. They identified what they call biogeographic stratification, which sounds like a geology term to me. What does that actually mean for a gut bug?
It means that geography is destiny for these microbes. A strain of BI living in a baby in Bangladesh is genetically distinct from a strain in Ghana, which is completely distinct from a strain in the UK. They mapped out 9 very specific geolineages globally.
So you literally have South Asian lineages, East African lineages, European lineages.
Yes. Because they are passed down vertically from mother to child, they evolve locally, they suffer from isolation by distance, they don't travel, they stay within a specific human population, evolving right alongside the people who carry them.
Which means if I had any BI left in my gut, it would look genetically different than someone's from halfway across the world. But why does that matter? Is it just random genetic drift, or are they actually doing different jobs?
That is the pivotal question of the whole paper, and the answer lies in their metabolism. This brings us to the actual superpowers of these microbes. We care about the difference between BI and BL and between the different regional strains because they eat fundamentally different things.
It's all about the menu.
It's all about the menu. BI is the ultimate milk specialist. Its genome is absolutely packed with genes designed specifically to break down human milk oligosaccharides, or HMOs.
And for the listener, HMOs are these highly complex sugars found in breast milk that the human baby actually cannot digest on its own.
Right. The mother is expending huge amounts of energy to produce sugars specifically to feed the bacteria, not the baby. And BI loves specifically fucosylated and cellulated HMOs. But there is a detail here that is so crucial, and you picked up on it when we were prepping. It isn't just about sugar. BI recycles nitrogen.
The nitrogen recycling mechanism. This is the part of the paper that completely blew my mind. Walk us through exactly how this works.
So breast milk naturally contains urea. Now, typically, we think of urea purely as a whaled product. It's what we excrete in urine.
Right, it's garbage to the human body.
But BI has this highly specialized urease gene cluster. It takes that urea waste, breaks it down, and releases the nitrogen trapped inside. It then recycles that raw nitrogen to synthesize its own amino acids and build its own proteins.
So it literally takes the baby's waste product and turns it into biological building blocks.
It is incredibly efficient. But here is the massive payoff for the human host. Because of this efficiency, BI operates as a localized vitamin factory. The study found that BI genomes are heavily enriched with genes to synthesize essential B vitamins, specifically riboflavin, folate, and biotin, and it utilizes pantothenate.
And what do those specific B vitamins do in a 2-month-old infant?
They drive neurodevelopment. They are the literal fuel for building a healthy, growing brain. So the bacteria eats the indigestible milk sugars and the urea waste, and in return, it manufactures the exact vitamins the baby's brain needs to develop. It is a flawless symbiotic loop.
That is just biological poetry. But now, compare that highly tuned machine to B. longum, the generalist that is taking over Western babies.
When they analyzed the BL genome, it was a completely different story. It was not enriched for milk digestion or urea recycling. It was heavily enriched for genes that break down plant glycans, things like arabinose, xylose, and fructose.
Plant fibers. So BL is basically gearing up to eat a salad.
Exactly. BL is biologically geared for an adult diet, or at least a weaning diet of solid foods. BI is engineered exclusively for breastfeeding. So when we lose BI in the West, we aren't just swapping one bug for another. We are completely losing that specialized milk processing machinery and the vital brain-building vitamin production that comes with it.
And this connects beautifully back to those 9 geolineages we just talked about. Because the study found that the BI strains in Bangladesh aren't just genetically different for no reason. They actually eat different things than the BI strains in Africa.
This was my absolute favorite part of the paper. It's the story element. They looked at the specific metabolic genes in these regional lineages. The BI strains native to Bangladesh are highly adapted to break down starch.
Which makes total sense because rice is the staple crop there. Even if the newborn isn't eating rice directly, the entire environment in the mother's diet is rich in it.
Exactly. But then look at East Africa. The BI strains isolated there are adapted to eat cellobiose and utilize vitamin C, which perfectly matches the local agricultural crops like teff, maize, and millet.
In West Africa.
In West Africa, the localized strains are enriched for vitamin B12 metabolism, which happens to perfectly match the specific nutrient profile of fonio millet, the staple grain there.
That is just wild. So the bacteria haven't merely adapted to human breast milk in general, they have adapted over centuries to the mother's specific regional diet and the agricultural history of that exact population.
It is profound. It suggests a deep biological coevolution, not just with human beings, but with human agriculture and culture.
Okay, that was a really dense section. Let's do our second checkpoint and recap before we move into how we actually solve this problem.
Right. So first, we have a stark metabolic divide. BI is the milk specialist. It eats HMOs, recycles urea, and makes brain-boosting vitamins. BL is the plant generalist, waiting for solid food. Second, in the West, we have largely swapped the specialist for the generalist, meaning our babies are missing that critical vitamin production. And third, globally, BI is not one size fits all. We have specific geolineages that evolved localized genetic tools to perfectly match local staple crops like rice in Asia or millet in Africa.
Now, this brings us to the really practical part of the deep dive. If BI is so crucial, and we in the West are entirely missing it, why is this even a problem? I can drive to the pharmacy right now and buy a probiotic pill that says Bifidobacterium infantis right on the label.
You certainly can, but the genomic data from the study suggests that what you are buying is essentially a lab rat.
A lab rat. That does not inspire confidence. What do you mean by that?
Think about the origin of these products. Most commercial BI strains on the market today, including the very famous ones like EVC001 or the scientific type strain DSM-20088, they were physically isolated from babies decades ago.
How long ago are we talking?
We're talking the 1950s and 1960s, and they were collected from infants in the US or Germany. So they are very old, highly westernized strains, and they have been living inside a sterile petri dish in a laboratory for 70 years.
Wow.
The study sequenced these commercial strains and found that they are highly clonal, meaning they are all almost genetically identical to each other. But worse, they are severely degraded. They have physically lost key functional genes.
Because of the genome reduction we talked about earlier, use it or lose it. If they aren't in a baby's gut, they throw away the tools.
Exactly. Living in a lab broth, they don't need to digest complex human milk. So many of these commercial strains are entirely missing specific HMO utilization gene clusters. For example, they often lack what's called the H1 cluster, which is absolutely vital for eating milk sugars. Or they are missing the EMNA pathway, which is required for N-glycan utilization. They are quite literally a biological shadow of the wild bacteria.
So we are basically trying to reseed a complex rainforest using a fragile potted plant that has lived indoors under fluorescent lights its entire life.
And the clinical proof is right there in the data. The study references recent probiotic trials conducted in Bangladesh. When doctors gave these Western lab-grown commercial strains to Bangladeshi infants, they utterly failed to engraft.
They couldn't colonize the gut at all.
No. They were immediately outcompeted and flushed out by the local wild strains of bacteria because the commercial strains simply did not have the right genetic tools to survive in a real-world, non-Western gut ecosystem.
Now, the researchers did find one local strain that worked better, right?
Yeah, they did. In previous work, they isolated a Bangladeshi strain called bug_2D9, and it performed much better than the Western commercial strains. But this new genomic atlas revealed that even bug2D9 is actually a genetic outlier. It is not fully representative of the broader South Asian BI population.
So even when we try to find a local solution, we might be picking the wrong candidate because we haven't mapped the whole territory until now. And there is a serious safety angle to all of this too, isn't there?
There absolutely is. The FDA has recently raised severe concerns, including warnings about sepsis in highly vulnerable preterm infants who are given commercial probiotics. When you are introducing live, potentially genetically degraded or environmentally ill-fitting strains into a premature baby's fragile gut, safety has to be paramount. This new global atlas finally provides the genomic reference map we need to rigorously screen these strains to ensure they are actually safe and functional.
So the current commercial probiotics are largely inbred, genetically weak, and they don't speak the local metabolic language. How do we actually fix this? This study proposes a whole new toolkit. And it actually starts with how scientists even search for the bacteria in the first place.
Right. It starts with the diagnostics. For years, scientists have been using PCR primers to detect BI in stool samples. You can think of a PCR primer as essentially a Control-F search function on a computer, but for DNA.
You type in the code you want, and it searches the genome to see if it's there.
Exactly. But this massive atlas revealed that the scientific community has been using a deeply flawed search term for a decade.
What was the error? What were they searching for?
The old primers were specifically targeting those HMO utilization genes. But as we just discussed, those specific genes are highly variable. Some regional strains have them, some have lost them, some use different pathways entirely. The study found that using those old primers resulted in up to a 55% false negative rate.
55%. That is staggering. So more than half the time we thought BI was completely missing from a baby, it might have actually been there doing its job, and our test just couldn't see it because it was a slightly different regional variant.
Exactly. Or the reverse happened. We thought we were detecting BI, but the test was actually picking up BL. It was a diagnostic mess. So to fix this, the researchers designed 30 new, highly specific primer targets. And the most important one for BI now targets that urease gene cluster we talked about earlier.
Oh, the nitrogen recycling machinery.
Yes, because it turns out, while sugar preferences might change based on geography, the ability to recycle urea is highly universally conserved across all true BI strains. It is a non-negotiable core function. So these new primers are exponentially more accurate because they target a piece of machinery the bacteria always keeps.
So we finally have better diagnostics. But what about better actual treatments? How do we replace the lab rats?
The authors argue that the era of the one-size-fits-all probiotic is over. We need precision microbiome therapeutics, or geospecific probiotics.
Meaning you can't just take a strain isolated in Germany in 1960, drop it into a newborn in Kenya, and expect it to magically rebuild the ecosystem.
Precisely. You need a Kenyan-adapted strain for a Kenyan baby. And the most exciting part of this paper is that it's not just theory. As part of this massive project, the researchers successfully cultivated 36 brand new, genetically distinct geospecific strains directly from infants in Africa and South Asia.
So they basically built a seed bank of wild, robust, fully adapted local strains.
Exactly. These are the prime candidates for the next generation of precision therapies.
That is an unbelievable resource for global health.
Right.
Let's do our final checkpoint and recap for this section before we wrap up.
Okay, so the takeaways here are, first, current commercial probiotics are basically lab rats. They are old, clonal, and physically missing critical genes like the H1 or MNANA clusters. Second, because they are degraded, they fail to engraft in real-world infants, especially in developing nations, because they get outcompeted by wild strains. Third, our historical diagnostic tools were fundamentally flawed, missing up to 55% of strains because they targeted variable genes. The new standard targets conserve genes like urease. And finally, the future of this field is precision probiotics, cultivating local wild strains specifically for local populations.
So bringing this all back home to you, the listener, why should you care about any of this? Maybe you aren't a newborn in rural Bangladesh. Maybe you are an adult listening to this on your commute in Chicago or London.
It matters deeply because it finally explains the biological why behind the probiotic failures so many people experience. If you have ever spent $40 on a probiotic supplement, taken it for a month, and felt absolutely zero difference in your health, this is the exact mechanism why. We are trying to use generic, degraded tools to fix a highly specific, complex, localized ecosystem.
And I think it also highlights a philosophical shift. Healthy does not look the same everywhere. A perfectly healthy infant gut in Mumbai looks genetically and metabolically different than a perfectly healthy gut in Manchester. We really have to stop viewing the Western microbiome as the gold standard default, because the data actually shows the Western microbiome is the one that is severely depleted. It is the one that has lost its biological heritage.
That is the most profound realization of the whole paper. Industrialization, for all its benefits, has fundamentally severed the biological heritage we pass down to our children. We have broken the ancient chain of vertical transmission. Bifidobacterium infantis is the ultimate milk whisperer. It is absolutely essential for early brain development and for training the human immune system not to overreact. And in the industrialized West, it is effectively gone.
Which raises a really provocative thought that I want to leave everyone with today. If this specific microbe is fundamentally responsible for training the immune system in the first 6 months of human life, and we now know it is missing in roughly 98% of Western babies,
we really have to ask ourselves, what chronic diseases are we dealing with today? What explodes in childhood allergies? What massive rise in autoimmune issues are we seeing that aren't actually standalone diseases, but are really just long-term symptoms of this one missing microbe? Are we basically trying to build a lifetime of health on a foundation that doesn't exist?
A really sobering thought. And it leaves a direct question for you, listening. If you could reseed your own gut or your child's gut with an ancestral foundational microbe, would you trust a wild strain newly plucked from a non-industrialized population? Or would you stick with the safe, sterile, lab-grown version from the pharmacy?
It really is the difference between a zoo animal and a wild animal. One is predictable, sure, but the other is biologically robust. Which one do you actually want guarding your health?
Something to really think about. Thank you so much for diving deep with us today. We will see you next time.